Hello everybody, welcome to the week 5 review video. My goodness, this semester's flying by. A few things to talk about because of course we are back to the 2-lecture schedule this week. So a couple of topics to talk about. First, I want to reframe this week just a little bit, just to kind of bring all of the different stories that we're telling this week together under one umbrella. And then we'll review 3 main topics. Complementation, which seems to have been relatively clear in the lecture, but I'm getting quite a few questions from you about related things in the lab, so we'll certainly talk about complementation briefly. We will review epistasis, dominant and recessive epistasis. At first blush, this does seem like a complex concept, but once we review it and practice with it this week, I think you'll agree that it's easily something that you can master. And then, by far, the favorite topic this week for review, or in other words, the most confusing topic from your feedback from your lectures, is genetic maternal effect and cytoplasmic inheritance. 2 very weird things. 2 things that are very, very similar to one another in that they represent full inheritance from the mother, but for extremely different reasons. And we'll talk about the reasons and the mechanisms of both of these effects at the last part of the video. So first thing, again, to reframe this week, I like to say that this week can be retitled Things That Made Mendel Say, that's funny. Because every single one of the things that we talk about this week, no matter how different the mechanisms or the actual details of the story might be, they all share in common that at their surface, they appear to break Mendel's rules, but as we dig deeper into the true genetic mechanism of these phenomena, they actually adhere to Mendel's rules. So Mendel, of course, did his very simple experiments with his pea plants. He was doing pretty simple analyses with traits that were controlled by single genes. He was lucky. We made that point in the first video that he chose such traits. And everything kind of made sense, and he came up with a wonderful theory of inheritance, and that's the basis of transmission genetics today. But everything that we talk about this week are things that don't really seem to follow those rules, seem to be exceptions to Mendel's rules. They seem to be things that would make Mendel say, that's strange. But in fact, when we dig a little deeper, we see that we are following Mendel's rules, and that's what we'll talk about today. So first things first, complementation. What we know already is that if we have someone who shows a mutant, or in other words, a recessive phenotype, they have to be homozygotic recessive. And if we have another individual of the opposite sex who is also mutant and recessive, and mate them, we would expect 100% of their offspring to be homozygous recessive and to show that mutant recessive phenotype. That's classic Mendelian genetics. Have a recessive phenotype, mate it with a recessive phenotype, and get nothing but recessive phenotypes. That's Mendelian genetics. Now, complementation involves having someone, a parent, with the recessive phenotype, mating them with another parent with the recessive phenotype, and when you mate them, 100% of the offspring have the wild type or dominant phenotype. Now, that's not Mendelian genetics. That's something that would make Mendel say, what is going on there? And this is the basis of complementation. Now, for complementation to occur, we really need one thing to be so. In fact, noticing complementation tells us that something is so, and that something is that 2 genes control the trait. And I should say at least 2 genes control its trait. You can have more. So one thing that I want to use as an example here is brown hair. A kind of a common trait that we used early on in the semester. We insinuated that brown hair is controlled by a single gene. That's not the case. Most of our traits, most of human traits are controlled by multiple genes. And here's our first example. And it makes intuitive sense, right? So remember that all genes encode proteins. That's something that I know you learned in General Biology 1 and or 2. We'll come back to this after the first exam when we go into molecular genetics. But all genes encode proteins. That's all they do. And to be more specific, individual alleles encode individual types of proteins. So I want you to imagine that in order to get brown hair, you need one protein to make that brown pigment. Right? Makes sense. Can't have brown hair without brown pigment, and you need a protein to make that pigment. And then that brown pigment goes to a second protein, because remember, this brown pigment's being made in the follicle cell, in a cell on the scalp. So that second protein pumps that brown pigment into the hair, making the hair brown. So you got to make the pigment and pump it into the hair, and that gives you brown hair. Makes sense, right? In order to have your actual hair brown, it needs to be pigmented, and that pigment needed to make it into the hollow shaft of the hair itself, into the hair protein. And of course, since this is a protein and this is a protein, that means that we need genes for a protein to make the brown pigment, and we need a gene for a protein that makes the pump. Now we're all set up to do this. I want you to imagine that gene A codes for the pigment-making protein and gene B codes for the pump. And imagine that we have two mutant alleles for the A gene. In other words, we have an individual that can't make brown pigment. What type of hair, what color hair would you expect this individual to have? And I heard someone say it, blonde. No pigment in the hair is blonde hair. Colorless hair is blonde hair. Now I want you to imagine that there's a second individual who's homozygous recessive for this B gene. They make brown pigment, but it stays stuck in the surface cells of the scalp. It never gets into the hair. It's never pumped into the hair because there's no functional B protein. What color hair would you expect that individual to have? Someone said it again, blonde. Because again, the hair is colorless. Here the hair has no color, so it's blonde, and here the hair has no color, so it's blonde. The reason for the no color here is that the pigment's not being made, and the reason for no color here is that the pigment's not getting into the hair. So two different genes that need to work together to give us our dominant phenotype. So far, so good. So, of course, anyone who has one dominant allele, at least one dominant allele for the A gene, and at least one dominant allele for the B gene, is going to have brown hair, because that individual can make brown pigment and pump that pigment into the hair. Now let's do the cross. We have an individual who is homozygotic recessive for A. We already established that they're going to make blonde hair because they can't make pigment. But they're homozygous dominant for B. So they've got a working pump, but it doesn't really matter, does it? Because they're not making pigment in the first place. Blonde hair. And we mate this individual with another individual who is homozygotic dominant for the A gene. So they make the brown pigment. But this individual is homozygotic recessive for the B gene. So since they don't have a working pump, their hair is still blonde. We have someone showing the recessive mutant phenotype mated with someone who shows the recessive mutant phenotype. And if we stop for a quick minute, we'll realize that Mendel would predict that given this cross, now remember, Mendel is naive to genotypes. He didn't really know how the DNA worked. He was just studying inheritance. So if Mendel saw a blonde-haired individual mate with a blonde-haired individual, what would he predict? He would predict that all their children would be blonde. But if we do this cross, we see that all of the offspring will have a dominant A allele from this parent and a recessive A allele from this parent. And all of these individuals that are born in the F1 will have a dominant B allele from this parent and a recessive B allele from this parent. And every single one of those kids is going to have brown hair. So when we mate this blonde individual with this blonde individual, 100% of their offspring have the dominant wild-type phenotype because the A alleles here are complementing the mutations here. The B alleles here are complementing the mutant alleles here, and we are seeing complementation. So that's really what complementation is. It is, to say it simply, when what's broken in the alleles of one gene are fixed by the dominant alleles of another parent, but what are broken for the alleles of that parent are fixed by the dominant alleles of the first. For complementation to work, you must have two unlinked genes controlling a trait on two different chromosomes. So that's the essence of complementation. Second on our list is epistasis. Whew, I love epistasis. Epistasis problems are like puzzle problems. I really, really enjoy going through epistasis problems. So another thing that's going to make Mendel say, huh, that's strange, that's funny. So what is epistasis? Epistasis is when one gene controls another. And the controlling gene is the epistatic gene. And the gene being controlled is the hypostatic gene. You might remember from the video I had cute kittens here, where one kitten was dominating another to illustrate epistatic and hypostatic. So what do we mean by one gene controls another? Let's say that there is a gene A, and when A is dominant, birds tweet very, very fast. So a fast tweeting pattern in birds. And when A is recessive, birds tweet very, very slow. So there's a slow tweet when A is recessive. But let's say that the A gene is hypostatic. It is controlled. And the B gene is epistatic. The B gene controls the A gene. Now, sometimes that control is going to break down to the answer yes. In other words, the B gene is going to say, hey, A gene, go ahead and do whatever the hell you want. You want the bird to tweet fast. If you have a dominant allele, you want the bird to tweet slow. When you have a recessive allele, when B says yes, it is not exerting its control. When B says yes, it is allowing the A gene to express. But the other side of that is that the B gene can say no. The B gene can block the A gene completely, exert its control, because it is the epistatic gene, and when the B gene says no, in this case, in this example, birds can't tweet. They're mute. So then the question becomes, well, how genetically does B exert its control? And that gets down to the two different types of epistasis. We have dominant epistasis, and we have recessive epistasis. And these terms here, dominant and recessive, these terms represent when the epistatic gene says no. Now, what do I mean by that? If this situation here was controlled by dominant epistasis, this would be a non-tweeting mute bird. Now, what am I representing here? What I'm representing here is that, and I'll say mute, is that once there is a dominant allele present, excuse me, B is exerting its control because it's dominant epistasis. If it's dominant epistasis, B says no when B is dominant. And then these two dashes here represent the A alleles, which don't matter at all because once B exerts its control and that bird is mute, it doesn't matter if we have dominant alleles for A, recessive alleles for A. B is masking A. B is exerting its control. It's blocking A's expression. This is a mute bird. So this is a mute bird. 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 As long as B is dominant in dominant epistasis, that bird is mute. So then the question becomes, well, when isn't the bird mute? When do we get to see a fast or a slow tweeting bird? Well, if it's dominant epistasis, and B says no when it's dominant, when does B say yes? Oh, I love it. I heard it. B says yes when B is recessive. Dominant epistasis means B says no and exerts its control when B is dominant. When B says yes, it's recessive. So this is a fast tweeting bird. This is a fast tweeting bird. This is a slow tweeting bird. We see the expression of A when B is recessive because in dominant epistasis, B exerts its control when it's dominant. Now let's go to a different example. Let's go to, I don't know why I'm hung up on birds, but let's say that dominant A is a brown bird, and recessive A is a red bird, and B is epistatic to A, so A is controlled by B. When B exerts its control, it's saying no, and no in this case is going to be a white bird. But B also has the option to say yes and allow A to express. But in this example, let's say it's recessive epistasis. The name of the epistasis always refers to when the epistatic gene says no. So when are we going to have a white bird if it's recessive epistasis? Well, the name of the epistasis always implies when the epistatic gene says no. So a white bird is going to occur when B is recessive. So this is going to be a white bird. This is a white bird. This is a white bird. This is a white bird. In recessive epistasis, B says, oops, sorry, can't see that. In recessive epistasis, B says no and exerts its control. B blocks A when it's recessive because it's recessive epistasis. The name of the epistasis implies when the epistatic gene says no. So when might we have a red or a brown bird? Well, if B says no when it's recessive in epistasis, when does B say yes? B says yes when it's dominant. So once B is dominant in this example, A is going to get to do whatever A calls for. So this is a brown bird. This is a brown bird. This is a brown bird. That's a brown bird. That's a brown bird. The way that we would get a red bird is we would have to have recessivity in A and at least one dominant B allele. So this is a red bird. That's a red bird. That's a red bird. If B says no when it's recessive in recessive epistasis, then B is saying yes when it's dominant in recessive epistasis, and we only see A's effect when B is dominant if the recessive epistasis is in play. So that's a lot there. I know that's very similar to what we talked about in the lecture with the dogs, and we're going to have some examples to practice with in our challenge questions. So I encourage you to back this up again and watch it again if you need to. But really what's going to always let you into this kind of material, what's always going to make this material accessible to you, is remembering that core principle. The name of the epistasis tells you when the controlling gene is saying no, when it's blocking the other gene. In dominant epistasis, the epistatic gene blocks the hypostatic gene when it is dominant. In order to see the hypostatic gene, the epistatic gene needs to be recessive. In recessive epistasis, the epistatic gene blocks the hypostatic gene when it is recessive. In order to see the effects of the hypostatic gene, the epistatic gene must be dominant in recessive epistasis. That terminology will help you keep epistasis in check. So the last thing that we want to talk about today is genetic maternal effect and cytoplasmic inheritance. Now these two cases really show a lot of what happens in terms of maternal inheritance, how the mother influences the offspring. But the mechanism and the outcomes are subtly different, and so we want to make sure that we keep them separate in our mind and we understand them completely. So we have genetic maternal effect and cytoplasmic inheritance. Let's talk about cytoplasmic inheritance first, I guess, because in many ways it might be the simpler one to understand. So to go back to health class a little bit, every female cell that's an egg, every female egg cell has, it's basically a complete cell. It has a nucleus. It has a half genome in there, right? So there's 23 chromosomes in there. We talked a little bit about this in our first lectures too. It has an endoplasmic reticulum. It's got some Golgi. It's got a bunch of mitochondria. It's got its lysosomes. It's essentially a human cell with half a genome. That's what a female egg cell is. And I don't know how much in other courses you've talked about mitochondria. I'm sure you've heard about them in high school biology as well. You've probably heard that they are the power plant or the powerhouse of the cell. They make energy. They're responsible for ATP production. That's true too. Everybody's usually pretty familiar about that once they get to genetics. Some other things, though, that you might not have encountered, I'm sure some of you have heard of this, but maybe some of you haven't, is that mitochondria are actually remnant bacteria. So the quick version of the story is that a very, very, very long time ago in eukaryotic cell evolution, a proto or pre-eukaryotic cell gobbled up and engulfed a bacterial cell as a meal, but for reasons that aren't entirely clear, that eukaryotic cell did not digest the bacteria that it just ate. That bacteria just kind of remained inside the cell without being digested. And it did what bacteria do, which is it absorbed nutrients around it. In this case, it came from the cytoplasm, and it made ATP. But the ATP that the bacteria made actually began fueling and energizing the eukaryotic cell that had eaten it. And this gave that eukaryotic cell a major evolutionary advantage. It could grow faster, it had more energy to thrive and survive and to make it through harsh times. And evolution always favors survival of the fittest, so these eukaryotic cells took over, and that's all that exists on the planet anymore. We outcompeted the rest. So our mitochondria, our organelles, are actually almost free-living prokaryotes that are inside our own cell. Kind of skeevy in a way, but that's what it is. And this kind of mutual arrangement that was established millions of years ago persists. What our cells get from our mitochondria is energy, a ready source of ATP, of energy, which we need to survive. What the mitochondria get from us is protection. Since they're already inside our cells, ain't nothing else going to eat them. And so it's a mutual relationship. It's a symbiotic relationship, and it continues to persist to this day. So why are we talking about this now? Well, one of the side effects of this evolutionary story is that, of course, that bacteria that was eaten long ago, this ancestor of the modern-day mitochondria, had its own genome. It couldn't exist as a prokaryotic cell, as a bacterium without its own genome. And that genome of that bacteria still exists in mitochondria today. We refer to it now as mitochondrial DNA, but it is circular, as we would expect a prokaryotic genome to be. It encodes its own genes, its own proteins, and it is mitochondrial. This DNA only exists in the mitochondria. So, pretty interesting, right? That our own mitochondria, these organelles in our cells, have their own separate genome, and that genome is a remnant or a residual genome from that prokaryotic genome of that individual prokaryotic cell that was eaten long ago. And to go back to what I was saying before, those mitochondria, with all their genomes, are in the egg, in the mother's egg, along with Golgi and endoplasmic reticulum and lysosomes and peroxisomes and a nucleus with half a genome. That's the woman's gamete. Now let's talk about the mighty male gamete. The mighty male gamete is far less interesting. It has a hard sugar coat, it has half a genome, it has a motor, and it has a tail. And that's all it has. It has no Golgi, no lysosomes, no peroxisomes, no nucleus, nothing but half a genome. A sperm is nothing more than an efficient delivery device for a half genome package. An egg is a fully functional human cell with half a genome in it. So, of course, what we know is when the sperm fuses with the egg, the DNA cargo of the sperm is delivered, it makes its way to the nucleus, we reconstitute an entire human genome, and that human being develops into a baby and is born. But notice what happened. The Golgi, the peroxisomes, the lysosomes, and the mitochondria of this new zygote, of this brand new human being, are all exclusively maternal. The only thing the father donated is half a genome. That's it. All of the cellular components in this cell came from the mother. That's true for all of us. We are all operating on the copied versions of our mothers, Golgi and ER, and lysosomes and peroxisomes and mitochondria. Now, it's in the mitochondria that this becomes interesting, because I just told you that mitochondria have their own dedicated genome that we call mitochondrial DNA. And that means that if there's a mutation in this mitochondrial DNA, in our mothers, every single one of our cells is going to have that mutation as well. Because our mitochondria come exclusively from our mothers. Everything in our cell comes exclusively from our mothers. So if our mothers have defective mitochondria, we have them too. Our fathers don't give us mitochondria. Our fathers don't give us mitochondrial DNA. Our fathers can't fix or complement or offset a mutation like this one. And so it doesn't matter about the mitochondrial health of our fathers. If our mother's mitochondria are faulty or mutated, we inherit that all the time. And by we, I mean males and females, sons and daughters. It's 100% inheritance. I want this term to remind you of this story. These terms have meaning. Cytoplasmic inheritance. Cytoplasmic inheritance is meant to remind you that we inherit everything in our cytoplasm from our mothers. And the mitochondria float in our cytoplasm. So we inherit even our mitochondria from our mothers. And since our mitochondria have their own DNA, we inherit that DNA from our mothers. So let's imagine that we have, and I'm going to use pedigree notation here. We're not going to cover pedigrees for a couple more weeks, but just to make it clear, in pedigrees, males are squares and females are circles. So let's say the mother is mutant. She's showing the mutant phenotype for this mitochondrial disorder. There's that optical myopathy that we talk about in the class, Leber something or other, the blindness, the early onset blindness. That's a mitochondrial disorder inherited in this way. So let's say the mother has that. She goes blind early on in life. It's recessive, and it's a mutation. She mates with a man who also has that same trait. And they have a son. And the son has that trait. And that begins to look X-linked, right? Where all the sons are going to get the mutation from the mother. But then they have a daughter. And the daughter has that trait as well. Well, that's not sex-linked, because if dad is healthy, dad should be giving a dominant allele. No, what we're looking at is cytoplasmic inheritance. Every single daughter and every single son born of this woman is going to have this disorder, because they all have only her mitochondria, and it's her faulty mitochondria that are causing this disorder. Furthermore, as long as these sons mate with a healthy woman, none of their children are going to have this disorder, because those children are going to get all of their mitochondria from whom? From the healthy mother. He doesn't pass his mitochondria on to his offspring at all, so he can't pass the disorder. By the same token, no matter who these daughters mate with, all of their children will continue to have this disorder, because they will continue to pass on those faulty mitochondria. So cytoplasmic inheritance embodies traits that are passed along what we call a maternal lineage, where every mother will pass it to every one of her children, but all those daughters will continue to pass it to their children, and all those daughters will continue to pass it to their children. It'll go down the female line. So that's cytoplasmic inheritance. Now let's go over to genetic maternal effect. Genetic maternal effect is a little bit different. So we already know that if a pregnant woman abuses drugs or consumes alcohol, in fact, even if she gets a very bad flu when she's pregnant, it can create havoc for the developing fetus and create permanent long-term challenges for that person as they're born and they grow up to be an adult. And we know this intuitively because, obviously, when new life is developing within the body of a woman, in the womb of a woman, her own physiology and the chemicals that she exposes herself to will also expose the baby, expose the fetus, and potentially have consequences there. So I think we intuitively understand that a woman's body, when she's pregnant, provides an environment for the developing fetus. What we sometimes don't understand or initially appreciate is that the proteins that the woman makes also affect the fetus. In fact, it's often maternal or mother proteins that set up the early body axis. The body axis is that the head is at the top, the rear end is at the bottom, the hands are at the ends of the arms, the back or spine is on the back surface, the lungs form on the ventral surface, and things like that. So those axes are set up by maternal proteins, believe it or not. And where do those proteins come from? Well, think back to what we talked about in complementation. Those proteins come from genes. So in many ways, just like alcohol consumption or drug abuse can affect a fetus's environment and development, so too can the maternal genotype, because it dictates the proteins that she makes, and those proteins have a direct result on the developing fetus. So that's really the crux of genetic maternal effect. In genetic maternal effect, the offspring, phenotypically, the offspring show the phenotype of the mother's genotype. And let me show you what I mean. So let's imagine a woman or a mother who is recessive, and she mates with a male who is homozygotic dominant. So that's her mate. And genotypically, when they mate, what are their offspring going to be? Their offspring are going to be 100% heterozygotic. That doesn't change, because they get a dominant allele from dad and a recessive allele from mom. That's the genotype of all the offspring. And the offspring are also 100% recessive in their phenotype. Well, what? Wait, what? What was that? Well, in genetic maternal effect, the offspring always reflect the mother's genotype. So these offspring are 100% recessive because they show the mother's genotype. Now, what I didn't tell you earlier is that this offspring here was born of a mother who is heterozygotic, who mated with a father who was homozygotic recessive. And she's recessive because she got one of his recessive alleles and she got her recessive allele. But if you look closely at this, she actually shows the dominant phenotype. This mother shows the dominant phenotype. Why? Because this encodes the dominant phenotype, and the offspring always reflects the genotype of the mother. The genotype of this mother calls for dominance, so she shows dominance, even though she's homozygotic recessive. Although she is showing the dominant phenotype, her homozygotic recessivity makes all of her offspring recessive. Now let's do the intercross, right? So let's take some of these F1s, so those are the females, and let's mate them with their corresponding F1 brothers. So we're doing that cross. And we already know the genotypic ratio, right? This is going to be a 1 to 2 to 1 ratio of homozygotic dominant to heterozygotic to homozygotic recessive. That's not going to change. And the phenotypic ratio is, and you're probably dying to say 3 to 1 dominant to recessive, but no. If it's genetic maternal effect, the offspring will always reflect the mother's genotype, and the mother's genotype here calls for what? Calls for dominance. So 100% of these offspring will show the dominant phenotype. In genetic maternal effect, the offspring always show the same phenotype. In other words, it's always 100% something, because the mother has a single genotype, and all of the offspring will reflect that genotype phenotypically. It's almost as if the phenotype lags a generation. The mom's genotype is calling for dominance. The daughter expresses it. This daughter's genotype is calling for recessivity. Her children reflect it. This mother's genotype is calling for dominance. Her children reflect it. So the children always show 100% consistency in their phenotype, and the phenotype they show is the phenotype that the mother's genotype calls for. That's genetic maternal effect. The reason why this is happening is because the mother's genotype dictates which kinds of proteins she can make, and those proteins are affecting the development of these offspring. In the lecture video, we talk about the coiling of the snail shells. So the mother makes one type of protein, and it causes the snail shells to coil clockwise. The mother makes another protein, and during development, it causes the offspring snails' shells to coil counterclockwise. Genetic maternal effect. The environment the mother creates with her genotype directly impacts the phenotype that the offspring can show. And so the offspring's phenotype is always correlated to the maternal genotype. All right, so again, commonalities between genetic maternal effect and cytoplasmic inheritance is this idea that the mother is directly influencing the offspring's phenotype with no input from the father at all. In cytoplasmic inheritance, that's because the mitochondria have their own genome, and that genome has its own traits, and those mitochondria come only from the mother, and since the father doesn't contribute mitochondria, the father doesn't contribute mitochondrial traits. In genetic maternal effect, it's because the offspring develop within or from the mother, so the proteins she lays down for development directly impact that. The father does have some say in genetic maternal effect, doesn't he? The father does have some influence, but it lags a generation. Let's go back to this. So here, dad is contributing nothing but dominant alleles. And the reason why 100% of these grandchildren, the reason why 100% of these offspring are dominant is because mom had this dominant allele which came from the grandfather. So there is input from the males in genetic maternal effect, but that input always occurs a generation later. It always occurs in the grandchildren because the direct descendants of that father are always going to phenotypically express the mother's genotype. So, things that make Mendel say, huh, all of these stories share that in common. Complementation, epistasis, genetic maternal effect, and cytoplasmic inheritance are all things that would make Mendel scratch his head and say, that makes no sense. But as we look deeper into it and we understand the biochemical and the molecular reasons for these stories, we understand that Mendelian inheritance principles are still being followed. We can still diagram this with Punnett squares. We can still predict offspring and infer parental genotypes and epistasis using what we know about Mendelian genetics. We just need to understand the mechanism of these phenomena, and we do. That's the whole point of the review video. So let me know in the comments if any of this is still confusing. We're getting pretty heavy and dense now, and understandably, we're nearing the end of the first unit. In fact, this week represents the end of Unit 1, so do feel fine expressing some latent or lingering confusion to me. That's all part of the learning process. But hopefully this did add some clarity for these topics as well. So until next time, of course, thanks for watching.